Thank you everyone for attending this symposium on lifespan neuroimaging, and thank you to the organizers for slotting me in to talk about some of our recent work on lifespan brain charts. Of course, we are by no means the first people to really try and delineate what typical and by extension atypical brain maturation may actually look like. There's been many much more eminent researchers before us that really have attempted to map out in detail what normative brain changes may look like in both structure and function. Here I'm just highlighting a few from Armin Rasnahan that has done quite a lot of work on sort of delineating these sex differential trajectories. And Nico Dozenbach, who's done a lot of work on mapping out what typical functional brain maturation may look like. And I think what's been quite influential in our research particularly is this very nice review paper from John Gilmore and colleagues from a few years ago, kind of outlining these particularly very rapid early changes in brain development. Now, despite the fact that there's already a lot known probably about how the brain matures and how the brain changes, there are still several challenges that really remain. So first of all, the age range of many prior models has often been restricted to, say, typically the sort of 6 to 30-year-old age range. And this is especially challenging in the context of developmental psychopathology, where it is well documented that many conditions have their onset much sort of prior to this range, which is very clearly sort of outlined in this review paper from a couple of years from Myers and Lee. But it's also challenging as it's well known that most of these sort of rapid changes in brain development happen very early on in development. Because most models likely don't really cover the onset of some of these key developmental windows. And in addition, perhaps many of these prior models tend to come from single, mainly cross-sectional data sets. And really, given the known variability of study and site-specific effects, as well as the potential for significant individual level variation, one really has to wonder how reliable that established norm really is. Now, me, not being a clinician or even a statistician really, my solution to all of these problems is usually to just try and get more data to try and fill some of these gaps in some of these known models. And this is really what we did. So first of all, we started with a very small team just mining publicly available data sets, such as the UK Biobank and ABCD and the various Human Connectome Project data sets. And then after we had a little bit of a sort of proof of concept and we had a very clear idea of sort of which parts of the lifespan we were really missing, crucial data effectively, we started reaching out initially people that we know, people that in our networks and people that have published really on some of these data sets, and asked whether they'd be willing to sort of help out and build this sort of normative reference point. And as you might have seen from the paper that was published, this is something that got out of control very quickly, to a point where I think we now have almost over 250 different people sort of collaborating and contributing to this resource. Now, lots of data is nice. So we're now at a point where we probably have around 150,000 structural MRI scans from slightly over 115,000 unique individuals. But we still thought it was very important to create a robust modeling framework first before really trying to create these normative charts for all of the possible phenotypes that you can extract from structural MRI. And even just looking at the raw data here, there are a couple of very clear and important modeling considerations that kind of have to go into that work. So as you can see in these sort of bell-shaped curves, you can, well, first of all, you can probably observe that a lot of the variation that you see is likely going to be nonlinear. It's not a clear sort of line going up and down. But you can also very clearly see that there's a varying level of variability. So if you see these plots, you see that sort of horn shape where the amount of variation between individuals becomes much bigger depending on where you are in the lifespan. And so this really forced us to think about sort of flexible modeling approaches. We wanted an approach that could sort of deal with that nonlinearity, that could kind of model that level of variation, as it might actually be a very important factor, but also one that doesn't make too many assumptions about the underlying distribution of this variability, just so we, in future, hopefully be able to scale this up to more fine-grained phenotypes. And then we also wanted a modeling framework that really provides the sort of population-level curves that allows for variable adjustment of things like site and age and all these other sort of technical covariates that likely come into play once you start aggregating data across different studies. And what we landed on was this framework called GAML-SS, which are generalized additive models for location, scale, and shape, which incidentally is also the modeling framework that the WHO recommends for using for generating normative growth curves. So I'm sure they didn't land on that by accident, and they had probably very similar considerations going into this. Now, using this method, we were able to generate these life-spanning trajectories going from about 15 weeks post-conception to really 100 years old, initially for the sort of 4 major tissue classes and then subsequently extend these to more sort of regional variation in volume as well as other morphological features, which is what you can see on this graph. So this is just looking at things like total cortical or average cortical thickness, total surface area, and sort of regional volumetric trajectories based on the desiccant atlas. In addition to that, it also allowed us in first instance really to determine some key developmental milestones that you might not be able to sort of measure throughout the entire lifespan using sort of other standard reference charts, such as those for height and weight. And I think what this really emphasizes is that like a lot of these key developmental milestones happen very early on in life. So say pre-year 6, like a lot of these sort of gross morphological features have already achieved their sort of maximum value as what they get throughout the lifespan. Crucially, though, our aggregated data set also contained like a large number of clinical cohorts. I think we have a total of about 110 different clinical labels. And so we could also use this reference charts to see whether specific clinical cohorts would deviate in any sort of systematic way from these typical trajectories. And perhaps unsurprisingly, we find that neurodegenerative conditions such as Alzheimer's disease and even mild cognitive impairment really exhibit like a very steep and skewed centile distribution, which is really confirming their relative reduction in specific tissues. And I think what's more striking even in this disease is that schizophrenia, even though it happens much earlier in life and might not necessarily be associated with big neurodegeneration, appears to exhibit a very similar level of deviation in these major tissue types. Whereas on the other hand, you see some of these developmental conditions such as autism or ADHD that show a much more subtle and much less homogeneous pattern. And to me specifically, I think that's quite an interesting thing to potentially follow up on, is where you can see that these distributions don't quite look normally distributed. So you can see, for example, here, you can see that there's a clear sort of bimodal distribution of some individuals that tend to be in the higher centile range and some individuals that tend to be in a much sort of lower centile range. But on average, they don't seem to deviate that much. And I think those could be quite interesting things to follow up on. Now, what this slide is also kind of emphasizing is that by virtue of doing this type of, let's call it normative modeling approach, where we're accounting for age, we can actually start doing sort of cross-diagnostic comparisons and really compare, for example, the effect sizes that we see in schizophrenia with those that we see in neurodegenerative disease. Now, another element I think that is quite important in this work is that, especially given that it takes, in our case, it takes about 20,000 sort of compute hours to generate these trajectories and really do all the sensitivity and sort of robustness analysis around some of these trajectories. We also wanted to create really a tool that would allow anyone else to run sort of centile scoring and as, if you will, benchmark their data set against our data set. And we did that by using this sort of maximum likelihood approach, which is really a way to sort of try and identify the various offsets in modeling parameters for a new data set and then compute centile scores from those. So we've tested this quite extensively now, and we currently say that you should probably have at least about 100 or so healthy volunteers to sort of estimate these offsets from, but then you can kind of propagate this to other data sets and really estimate centile scores for any new data set. So we've made this available online on our website. And I should stress that, like, if you're uploading data there to compute your own centile scores, we're not retaining any data, nothing is stored. We can't even see if people are uploading or just sort of browsing this website. So it's all also GDPR compliant, and we kind of hope that this can become a resource for people to sort of really use as they continue to benchmark their data against ours. And finally, I think there's a lot of sort of things that sort of lessons learned, and perhaps this is sort of a message to the community, perhaps to some extent, really just to sort of say that there's a lot of public data already available, let alone like data that's sort of clinically acquired. So one of the things we've seen is that we've been able to, with relative ease, like lots of Zoom calls and lots of emails, pull together like 100,000 or so scans. But this really pales in comparison to the number of sort of clinical brain scans acquired, and I think that's really going to be a key sort of area of growth to try and tap into some of that. We've also noticed that, as you can see from like all the people that have been involved in this project, that people are generally happy to sort of share simple data and really contribute to this. So if we're building these sort of simple reference charts, I don't think anyone is too hesitant in contributing to that and helping out. The other thing is, I think, hopefully what we've shown is that you can also actually learn quite a lot from just even very simple features such as sort of gross morphological changes, because we can already see some basic clinical differences in some of those measures. So we don't even need to go to like vertex level or voxel level differences to observe some of these gross clinical differences. Now, I think the main thing we learned from this project, though, there's a lot of room to expand. So I was showing earlier that slide where we see these different clinical conditions, and I think every little sort of asterisk denoting a significant case control difference is something that we could follow up in much more detail. Because if you remember some of those differences, even though we're presenting them as broad case control differences, they're really the underlying data, and that is sort of individual level centiles. And I think there's a lot of room to see whether there are specific subgroups in those data sets, such as we see potentially signs for in some of these developmental conditions, but also to see if this sort of level of deviation is related to specific symptoms or specific behaviors. And I think on top of that, we did a little bit of evaluation to see to what extent these models are stable when you have longitudinal data, and suffice to say they are very, very stable, but I think there's also a lot of room to sort of follow up to see like how some of these individual level deviations mature in longitudinal data. And then there's a lot of sort of ifs and buts. I think one of the main things that we realized is that actually this does require a community effort. Like no sort of single group would have been able to collect any of this data. So really all of this stands or falls with people really coming together and kind of pulling their resources. The other big caveat I think to put in any of this is that all the data we've gathered so far is still very largely restricted to sort of Western majority-wide, say, sort of North America, European ancestry. And even though we have some data from South America, from Africa, from Asia, Australia, etc., I think there's still like a clear bias in this data set, and that's something that we have to remedy by specifically targeting sort of traditionally underrepresented populations, especially if we ever want to sort of move this into a sort of a clinically translatable tool. And then finally, like none of this would be possible without like having a good sort of informatics IT infrastructure behind it, to the point I think we're now at a stage where we probably have about 1 petabyte of data and use well over 2 million CPU hours on this project. In an ideal scenario, we'd be able to sort of share all this process data publicly with everyone, but obviously that's not always possible. So a sort of next best option for us was to at least try and share these models and try and allow people to benchmark their data against ours. But hopefully in future we can work towards a model where data is more easily and more widely shareable, and everyone is not like redoing work that other labs have already done in terms of processing data. And so with that, I'd like to end and really thank all the collaborators, a list that's continuously growing, and the key team, and obviously the funders for making a lot of this work possible.